Now, we move on to specification 9, which asks us to investigate and describe the structure of a seed. Here, we focus on the main parts of the seed, the embryo, which includes the radicle, plumule, and cotyledons, and the outer protective layer called the testa. Studying the structure of a seed involves examining these parts carefully, often using a dissected seed for observation. So, we also have a practical work here which is needed for your ATP paper, so let's see. The general structure of a seed can be understood by observing both its external and internal parts. A seed is a specialized, dormant structure that contains a young plant called the embryo and stored food. It is protected by a tough outer covering and remains inactive until suitable environmental conditions allow it to grow into a new plant. In the external view, the seed is covered by a protective layer called the testa, or seed coat. This tough outer covering protects the delicate embryo from mechanical damage, pathogens, and drying out. On the seed coat there is a scar called the hilum, which marks the point where the seed was once attached to the fruit wall or pod. Near the hilum there is a very small opening called the micropyle. This tiny pore allows water and oxygen to enter the seed, which helps initiate the process of germination. In the internal view, the main structures visible are the cotyledons and the embryo. The cotyledons, also known as seed leaves, store food such as starch, proteins, or fats that provide nutrients for the developing seedling until it can produce its own food. A seed may have one cotyledon in monocot plants or two cotyledons in dicot plants. The embryo is the young plant present inside the seed. It consists of the plumule and the radicle. The plumule is the embryonic shoot that later develops into the stem and leaves of the plant. The radicle is the embryonic root and is usually the first part to emerge during germination. Germination is the process in which the embryo begins to grow after a period of dormancy. First, the seed absorbs water through the micropyle, causing it to swell and the testa to soften and break. Then the radicle emerges and grows downward to anchor the plant and absorb water and nutrients from the soil. After that, the plumule grows upward toward the light and develops into the shoot. During the early stages of growth, the cotyledons supply food to the young seedling until it can carry out photosynthesis and grow into a fully developed plant. Again, we start looking at the structure of a seed from fertilization. After fertilization, the ovules develop into seeds. Each seed contains a zygote, which is the fertilized egg cell. This zygote divides and grows to form the embryo, the young plant inside the seed. The embryo is surrounded by cotyledons, also called seed leaves. Depending on the type of plant, a seed may have one cotyledon in monocots or two cotyledons in dicots. These cotyledons store food, providing energy for the growing embryo until the seedling can produce its own food through photosynthesis. When conditions are favorable, the seed absorbs water, which softens the seed coat, called the testa. This allows the radicle, the first root, to grow downward into the soil, and the plumule, the first shoot, to grow upward toward the light. The embryo then develops into a young seedling, starting the growth of a new plant. This structure ensures that the embryo is protected, nourished, and ready to grow into a healthy plant when the environment is suitable. Also, the seed is covered by an outer protective layer called the testa, which shields the developing embryo. Inside, the embryo contains the plumule, the embryonic shoot, and the radicle, the embryonic root. The cotyledons act as a food store, supplying nutrients and energy to the seedling during germination. The seed coat also has a tiny opening called the micropyle, which allows water to enter and start germination. As the seed grows, the plumule develops into the shoot of the plant, while the radicle grows downward to form the root system. From the outside, the seed shows the protective testa, and when viewed internally, the cotyledons, embryo, plumule, radicle, and micropyle can all be seen, each supporting the development of a new plant.